In this lesson, we're going to focus more on the process of converting DNA into RNA and protein, not necessarily looking at the nitty-gritty details at the cellular level, but how is this transfer of information from the nucleus possible as it goes through RNA to protein? Well, let's go back to the central dogma of biology. Remember that Francis Crick in 1956 provided this very important central theme that DNA is responsible for the production of RNA. The RNA leaves the nucleus and goes to the cytoplasm, and eventually that RNA has to be read in a way that produces proteins. And those amino acids must be laid down in a very particular fashion. So that is the central dogma. Let's take a little bit of a closer look at RNA. And it turns out there are three main kinds that we need to be concerned with. And let's take a look at them in turn. Firstly, RNA, like DNA, is a carrier of genetic information. It's a little bit like the sacrificial lamb in a way, because DNA never leaves the nucleus, and RNA does. And when it leaves the nucleus, it's a little bit more vulnerable to things that can degrade it than it would be in the nucleus. Let's look at the key differences between DNA and RNA. You'll remember that DNA contains a deoxyribose sugar, and RNA contains a ribose sugar. So one small difference, there is a hydroxy group that's present in RNA in the 2' position that is not present in DNA. Another key difference between the two is that DNA is double-stranded. We know this from the Watson-Crick model, whereas RNA is single-stranded. It turns out that single-stranded RNA is way more susceptible to being broken apart than DNA. The 4 bases in DNA, remember, are adenine, thymine, cytosine, and guanine. Another key difference in RNA, of course, is the replacement of thymine for uracil in RNA. So those are the 3 key differences between the two. Now let's look individually at the 3 kinds of RNA that we'll be looking in more detail in this course. Firstly, we have messenger RNA. This is the RNA that has been in the news a lot lately. In fact, if you're following, both Pfizer and Moderna vaccines are produced using messenger RNA. So this is a very timely topic. In a nutshell, messenger RNA, or mRNA, takes the message from the DNA in the nucleus and carries that message into the cytoplasm where protein synthesis will ultimately occur. And you'll remember that the ribosomes are really important in terms of producing protein. The ribosomal RNA, or rRNA for short, is part of the machinery that actually binds to the proteins and the ribosomes. So we'll look at this in more detail in a future lesson. Transfer RNA, or tRNA, has a very recognizable shape to it. It's a clover-like structure, and it works very closely with ribosomal RNA and the ribosome to actually read the message, which is the messenger RNA, kind of like a ticker tape, and ultimately forms part of the machinery to make protein. And we'll look at this in a lot more detail as well. So a quick overview of transcription is in order here. We'll also take a look at translation. Remember, transcription is really the production of RNA from the DNA, and this kind of happens in the nucleus before the RNA leaves. An enzyme called RNA polymerase is responsible for reading that DNA molecule and producing the RNA that comes from it. Since DNA is read from the 3' to the 5' direction, the messenger RNA is always formed in the 5' to 3' direction. Very much like DNA when it was replicated, if you recall. The daughter strands are always produced from 5' to 3'. RNA is no different. The precursor mRNA, which is sort of unmodified, has to be changed a little bit before it's able to make protein. And we'll talk about this in more detail in a future lesson. Here is a general overview of what happens when we start from DNA and make RNA and finally protein. The top structure shows us the double-stranded DNA molecule, and we are going to actually transcribe the top strand. And remember, the DNA is always read from 3' to 5'. So we're going to focus on the top strand. The first 3 pieces of DNA that get read, or nucleotides, are T, A, and C. The complementary strands of RNA that are produced from that are A, U, and G. And that makes sense because thymine in DNA codes for adenine in RNA. Adenine in DNA, right here, codes for uracil in RNA. Remember, there is no thymine in RNA. And finally, the cytosine up here in the DNA molecule ends up being transcribed into the guanine in the RNA molecule. So it's quite simple in a way when you look at it. As RNA polymerase reads across this DNA, it lays down brand new RNA that's complementary from the 5' to the 3' position. The next step, called translation, remember, occurs in the cytoplasm, and this is where the amino acids are very carefully laid down using the ribosomal RNA and the tRNA when they read the messenger RNA. And we'll look at this in more detail, but it turns out that methionine is always the first amino acid to be laid down. And after that, the messenger RNA simply gets read with triplet codons. That means 3 bases in a row, and each of those triplet codons results in a very specific amino acid. At the end of the day, this is how we make protein, and those proteins, of course, produce the traits that we call phenotypes. So that is a good overview of transcription and translation. So let's take a little look at the genetic code. There must be a genetic code in the DNA that codes for each of the 20 amino acids. That was clear from Beadle and Tatum's experiments. The problem that arose in the 1950s and a little bit later was simple. How can 4 single nucleotides, A, C, T, and G, code for 20 amino acids? There's a little bit of a problem here because one nucleotide certainly cannot code for enough amino acids to make all the proteins in our body. If each code or codon were made up of 2 bases, such as A and G, there would only be 16 codons. I can show you more in detail about what I mean here. If you think about it, if we have 2 different nucleotides involved, we can have 4 possibilities in this position right here, A, C, T, or G, and we can only have 4 possibilities here. Well, when you multiply those 4 together times 2, that still only represents 16 different codons. And of course, because there's 20 different amino acids, there would be less than 1 codon available for each amino acid, which seems to be impossible. If each codon, on the other hand, is made of 3 different bases, we would have enough codons to easily cover the 20 amino acids plus a bunch of extras. Interestingly enough, we only need 20, but we would have 64. The reason for that is mathematical. We have 4 possibilities for the first codon position, A, C, T, or G, multiplied by 4 in the second position, A, C, T, or G, multiplied by 4 possibles in the third position, A, T, C, or G, and that would give us a total of 64 combinations of A, C, T, and G, more than enough to code for each of the amino acids because there's only 20. Well, this is really what gives rise to the genetic code. It turns out that each codon does consist of 3 nucleotide bases. An example of a codon would be AUC in the RNA. Let's look at the genetic code a little more closely. Firstly, there is replication in the code, or redundancy. It turns out that many different amino acids have greater than 1 codon that code for them. If we look at leucine, for example, it has 6 different codons that code for the same amino acid. This is where we see this right here. There are 6 different codons that code for leucine: UUA, UUG, CUU, CUC, CUA, and CUG. This actually produces a lot of protection against mutations because of the redundancy in the code. Secondly, the genetic code is unambiguous. Each triplet codon has only one meaning. That means that if we're going to look at this codon right here in the top left corner that codes for UUU, that codon only codes for the amino acid phenylalanine. It does not code for another different amino acid. Finally, very importantly, there are stop codons that are built in and one start codon. The start codon is always the same. It is AUG. And like I've already said, the start codon always codes for methionine, which is one of the 20 amino acids. On the other hand, we have 3 different possibilities for stop codons. When we get to the end of a protein, somehow we have to stop making amino acids. And the genetic code is set up in a very interesting way that shows the stop codons in red right here. If a triplet codon with the order UAA, UAG, or UGA in the RNA are encountered, then that will be the end of the protein. And the machinery will fall off of the protein, and that protein is ready to go and perform its function in the body. So if we look at the big picture, we can start with DNA and we can figure out what the messenger RNA would be from that. So here is our DNA molecule. We have the 3' end on the left and we have the 5' end on the right. So I've picked a very short segment of DNA just to give us an idea. When that DNA is transcribed, obviously we make the RNA gets transcribed from left to right, in this case, 5' to 3', and you can see the order. It'll be CGG, UAC, UGG. That is transcription. Once that RNA leaves the nucleus, then it can be translated into protein. When we look at the genetic code, this triplet right here, CGG, codes for a specific amino acid, and that amino acid is arginine. The next triplet codon, UAC, codes for another specific amino acid, and according to the code, that would be tyrosine. Finally, the last triplet codon that we've got in this very short segment of messenger RNA would code for tryptophan. So that, my friends, is a very quick look at translation DNA converting to RNA, transcription, DNA going to RNA, and translation, RNA going to protein.